Hello, my name is Victoria Yang.
And my name is Moosikasuwan, and we study the remediation of amyotrophic lateral sclerosis pathogenesis with autophagy inducers and antioxidants on the C. elegans. ALS is a motor neuron degenerative disease, damaging motor neurons in the muscles, leading to muscle atrophy. It affects thousands of patients and is expensive to treat.
Figure 3 displays the mechanism of action of ALS at the cellular level and how mutated cells and aggregates give rise to the illness. Misfolded TAR DNA binding protein 43 can form aggregates and impair mitochondrial function. This is dangerous because mitochondria manage ATP production and intracellular signaling.
Figure 5 displays the role of oxidative stress in ALS. Free radicals known as reactive oxygen species cause mutated proteins and aggregates and impaired mitochondrial function. Figure 6 describes the mechanisms of autophagy. Autophagy can remove toxic proteins that are attributed to neurodegenerative diseases and prompt the regeneration of healthy cells.
A study by Yoon et al. In 2018 determined that trehalose activated the most autophagosomes in each cell. Autophagosomes are the key structures of macroautophagy, so trehalose would be the most effective at inducing autophagy and removing mutant cells and protein aggregates. Sesnick et al. In 2020 examined the mechanism that TDP-43 takes to form aggregates that cause motor neuronal death. In standard situations, TDP-43 forms small aggregates that are easily removed, but when the gene is mutated, it forms larger aggregates causing disturbance of neuronal activity.
The purpose of the study was to determine if trehalose, an autophagy inducer and antioxidant, could reduce ALS-related neuron death and increase motility in C. elegans. We predicted that when exposed to the highest concentration of trehalose, C. elegans strains would exhibit greater motility, lower concentrations of TDP-43, and increased cholinergic neuron activity.
The studied strains of C. elegans were BH254 to monitor tauopathy and progressive neurodegenerative disorders, LX929 with GFP expression in cholinergic neurons, and CL6049 with GFP expression in TDP-43 intestinal neurons. C. elegans were age synchronized and treated with 0, 1, 5, or 10 millimolar of trehalose. Body fashion and paralysis assays were conducted. Fluorescence microscopy assay was performed to compare the extent of cholinergic neuron death and TDP-43 presence in worms with and without treatment, and data was analyzed with a one-way ANOVA and post-hoc Shefa using IBM SPSS version 26. Figure 10 displays the body thrashing assay, which was done by placing a drop of M9 on an unseated NGM plate with a single nematode and recording the body bends for 60 seconds.
Figures 11 and 12 display the data that was collected and analyzed. Significance between the various concentrations groups are denoted by the symbols in the key. Figure 11 displays the body thrashing data. Significance is present between the LX99 10 millimolar groups and the CL6049 control groups. Figure 12 displays the paralysis data. Significance is present between the LX99 control and the 1 millimolar and 5 millimolar groups, and between the CL6049 control and trehalose exposed groups. In both assays, the motility responses were dose-dependent, with the 10 millimolar performing the best. Figures 13 and 14 show the fluorescent microscopy conducted to examine the levels of cholinergic neurons in the LX99 strain. 13 shows the control, and 14 shows the vibrant neurons of the 1 millimolar treatment. We originally planned to quantify the concentrations of cholinergic neurons, which use acetylcholine to transmit chemical impulses in the LX99, and the concentrations of the mutant TDP-43 gene protein in the CL6049 strain. Fluorescent microscopy was a technique that we had challenges with, and we were unable to quantify the photos we took.
The purpose of this experiment was to observe the effects of the autophagy inducer and antioxidant properties of trehalose on ALS pathogenesis. Trehalose treatment improved motility as seen statistical significance between groups. There is a dose-dependent positive effect as the most effective concentration was 10 millimolar. Visually, there were more cholinergic neurons present in the groups treated with trehalose. The quantification did not occur due to complications with the fluorescence. Motility was likely improved due to the elimination of aggregates via autophagy, which allowed for a greater ease for neurotransmitters to send chemical signals like acetylcholine to create movement in the motor neuron cells of the nematodes. Trehalose is an antioxidant, allowing it to donate electrons and neutralize free radicals like toxic tau aggregates. Barber and Shaw in 2010 determined that oxidative stress is present in ALS patients, potentially due to mutations in the superoxide dismutase 1. Mizuno et al. In 2018 determined that trehalose successfully mediated oxidative stress in mice, suggesting trehalose antioxidant properties. Trehalose is also an autophagy enhancer, stimulating phagophores, which are double membranes that enclose the mutant proteins, which are the TDP-43. They are then isolated into an autophagosome and degraded by the autolysosome. Sheena et al. 2009 determined that TDP-43 is a dimeric molecule, meaning that it is an oligomer composed of smaller versions of itself. It synthesizes at the N-terminal, which is the free amine groups at the ends of the peptide. Therefore, its homodimerization assists in the aggregation of the proteins. And furthermore, Zhang et al. In 2009 demonstrated that the first 10 groups of the N-terminal dimerization were involved in the aggregation of full-length TDP-43.
In summary, we determined that trehalose could potentially remediate ALS pathogenesis, as trehalose treatment resulted in increased motility in the nematodes, implying a decrease in neuron death. Therefore, autophagy inducers with antioxidative properties like trehalose could potentially remediate ALS pathogenesis. We hope to enhance our understanding of this topic by studying its efficacy on other ALS pathogenesis pathways, like the SOD1 or glutamate excitotoxicity, exploring other neurodegenerative diseases, or using other antioxidants. This is our bibliography, and thank you for watching.